Hello, everyone, and welcome to today's live broadcast, Looking Beyond PCR, Isothermal Amplification. Our webinar is presented to you by Dr. Agne Alminaite and Dr. Remigijus Skirgaila. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please click on the sponsor logo on the left side of your screen. Now, before we begin, I would like to encourage you to get the most out of today's webinar by submitting as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We will answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or use that Ask a Question box on the left. This presentation is educational and thus offers continuing education credits. Following this presentation, those credits can be obtained by clicking on the tab at the top right and following the process to receive your credits. I would now like to introduce today's speakers. Dr. Agne Alminaite, Product Manager, Custom Molecular Biology Products Management Group, Thermo Fisher Scientific, and Dr. Remigijus Skirgaila, Lead R&D Manager, NGS Library Preparation and Protein in Vitro Evolution Group, Thermo Fisher Scientific. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Dr. Alminaite and Dr. Skirgaila, you may now begin your presentation.
Thank you for a nice introduction. Hello to everyone, and hello to everyone interested in what isothermal amplification can offer for your research. So today our topic is looking beyond PCR and isothermal amplification. Today we'll talk in general what kind of group of techniques fall under this common term of isothermal amplification, and we'll concentrate more on the multiple displacement amplification, or MDA, and its importance for whole genome amplification, and as well the story of the development of one unique ECVFI 29 DNA polymerase. Another isothermal amplification technique that we will discuss is loop-mediated isothermal amplification, or LAMP. And finally, we'll give some insights on the applications of these techniques for diagnostics. So let's start from the term itself. Iso means equal, and equal means that these kind of reactions, isothermal amplification reactions, can occur at the same temperature all the time. This makes them very convenient, and at many cases eliminates the need for expensive equipment. In general, nucleic acid amplification is a technique used to amplify starting material, and it is pivotal for all types of research. In life sciences, in diagnostics, in agriculture and forensics, we will find these techniques being used. Because of these characteristics, isothermal amplification offers many advantages over PCR. As I mentioned, no expensive equipment is needed, the chances of contamination are reduced, and these type of enzymes are not so sensitive to reaction inhibitors, and in many cases, tests that are out of laboratory environment can be developed, and such tests can be used then in field situations or point-of-care tests. Over the years, there has been a list of enzymes discovered that can carry out these type of reactions. So the list of them has been growing, and so has been growing the methods that were developed using these kind of enzymes. And depending on the starting material, would it be DNA or RNA, the enzyme that carries out the amplification, or other factors in the reaction, there has been a multitude of isothermal amplification methods developed. So you just have to pick the one for your needs and keep amplifying. The number itself of the enzymes for isothermal amplification is long, but also the applications and the techniques where these kind of methods are used has been growing. Global market of applications has been increasing since the development of these techniques in the early 2000s. These methods are used in many areas of research and diagnostics. We will find them in blood screening, for example, to detect SNPs or small nuclear repeats to track the inheritance of disease genes. They are also used for infectious disease diagnostics, both directly in point-of-care facilities or even at home, and also in hospitals. Also, the use of such techniques are growing in cancer research, for example, for cancer-specific molecular biomarkers detection. And as today we can see, the area that's been growing most probably is infectious disease diagnostics, where these tests using these isothermal amplification techniques become very important. Out of all the isothermal methods that we have and that have been developed to date, now we will talk about two. And you may ask why choose these two methods? We think they represent the two very important research areas. So first of all, we'll talk about the MDA, multiple displacement amplification. And MDA is mostly used in genome studies. This type of technique is very versatile. It offers extensive genome coverage, which is very important in this field of research. It gives us high DNA yields from a very small amount of starting DNA material. Starting DNA material can be even a single cell. MDA offers low amplification bias, which is one of the main pain points of the researchers doing this kind of studies. And what enzyme is behind all of this method is Phe29 DNA polymerase. Another method that we are talking today about is LAMP, or loop-mediated isothermal amplification. And we chose this method to talk about today because it's one of the most important methods for rapid pathogen detection. This method offers a very simple reaction setup, fast results. It can be used in the field conditions, and because of the polymerase that carries out of this reaction, the BSM or BSD polymerase, such reactions are very inhibitor tolerant. So even crude DNA or DNA samples can be used as the starting material. Okay, this was the introduction, and I'd like to hand it over now for my colleague Remigijus to talk more about MDA and whole genome amplification.
Thank you, Dr. Alminaite. Now, before we pass it over to Dr. Skirgaila, we would like to ask our audience our first question. Are you currently or planning to perform whole genome amplification? Simply check the yes or no box, and we appreciate your participation. Okay, Dr. Skirgaila, you may now begin your presentation.
Hello, everybody. Thank you, Agne. So my name is Remigijus Skirgaila. I'm about to continue our webinar. At this point, I would like to cover whole genome amplification using multiple displacement amplification technique driven by Phe29 DNA polymerase and special development of Thermo Fisher Scientific Equi-Phe29 polymerase designed for more efficient MDA reaction. First of all, a few words about multiple displacement amplification technique. It is used for non-specific DNA amplification. The MDA is performed on single-stranded DNA by annealing of random polynucleotides. Key enzyme in this process is Phe29 DNA polymerase, which elongates primers and complementary DNA strands is synthesized. At the moment the polymerase encounters double-stranded DNA region, it starts to displace upstream DNA strands. The displaced DNA strand, being single-stranded, is a perfect template for new priming events. As the MDA reaction continues, new DNA strands are synthesized, generating hyperbranched DNA structure, which preserves original genetic information of template DNA. Once again, the key enzyme in multiple displacement amplification is Phe29 DNA polymerase from Bacillus subtilis stage, which has an exceptional strand displacement activity and processivity of up to 40,000 bases per single binding event. MDA, driven by random priming and Phe29 DNA polymerase, could easily amplify starting DNA amount more than 10,000 folds and generate enough of material for all subsequent applications. DNA amplified by WGA could be used in many different downstream genetic analysis applications like Sanger sequencing, next generation sequencing, genotyping with microarrays, single nucleotide polymorphism, and copy number variation analysis. A number of research areas require DNA amplification. It could be gene banking, single cell analysis, genetic disease research, etc., etc. Single cell analysis is a fast-growing field which is looking for differences in cell population at single cell level. For genome and transcriptome heterogeneity analysis at single cell level, it is necessary to have an access specifically to one single cell. Cell separation could be done by dilution or using some specialized instruments like fluorescence-activated cell sorters. Nowadays, it is also very popular to compartmentalize cells in emulsions or microwells. Once the individual cell is accessible, its genetic information could be analyzed. And again, in some workflows, we need nucleic acid for amplification. Returning back to non-specific nucleic acid amplification, we could identify three different groups of amplification techniques. So it could be PCR-based, like degenerate oligonucleotide primed PCR or primer extension preamplification. B, hybrid techniques like linear amplification via transposon insertion, loyanti, or multiple annealing and looping-based amplification cycles, now back, and finally, again back to multiple displacement amplification. Specifically for multiple displacement amplification, Thermo Fisher Scientific has designed Equi-Phe29 DNA polymerase. The enzyme was developed using in vitro evolution technique, which we name isothermal compartmentalized self-replication, ICSR. The gene of wild-type Phe29 DNA polymerase was cloned into a plasmid, and the enzyme was expressed in a Escherichia coli cell. The library of mutant variants of Phe29 polymerase was generated using error-prone PCR. Escherichia coli cells expressing the mutant polymerases were compartmentalized, and water in oil emulsions containing no more than single filter compartments were prepared. Escherichia coli cells were disrupted using lysozyme treatment and repeated cycles of freezing thawing. Released Phe29 DNA polymerase could amplify its own genome located in a plasmid by performing MDA at certain conditions. The screening for more efficient MDA enzyme was performed at increased reaction temperature and reduced amplification time. Final variant of DNA polymerase was identified after several iterative screening cycles. Newly developed mutant P29 enzyme was tested in MDA reaction comparing to wild-type enzyme at standard 30 degrees Celsius and increased 40 degrees Celsius reaction temperature. DNA amplification is presented as a number of amplification folds at different reaction times. The curves for mutant P29 DNA polymerase are in red and wild-type enzyme data points are in blue. For the first 2 hours, DNA amplification at standard 30 degrees Celsius, red and blue triangles, MDA temperature is similar for both enzymes. However, later on, wild-type polymerase slows down. Meanwhile, mutant P29 reaches about 30,000 folds amplification comparing to 10,000 folds for wild-type after 6 hours. Let's take a look at MDA reaction performed at 40 degrees Celsius, red and blue squares. At this temperature, wild-type polymerase is essentially unable to perform MDA reaction, blue squares. Meanwhile, mutant enzyme works at optimal temperature. MDA reaction is about 2 times faster, and about 30,000 folds amplification rate is achieved within 3 hours of reaction, red squares. Based on mutant polymerase variant coming from in vitro evolution approach, Thermo Fisher Scientific has launched commercial enzyme Equi-529. This enzyme was tested by performing MDA on FACS-sorted single cells from human cell lines or 6 picograms of human genomic DNA. The experiment was performed in 3 steps, workflow comprising cell lysis, neutralization step, and MDA performed for 1 hour at 42 degrees Celsius. Total workflow time is about 2 hours, which includes about 30 minutes of hands-on time. The key features of MDA performed by Equi-529 DNA polymerase are increased reaction temperature, higher reaction speed, better product yield, and reduced amplification. On this slide, GC bias of single-cell DNA preamplification is shown as a normalized coverage curve for sequenced human genome regions of different GC percentage. An ideal coverage curve would be flat line at 1 all over the GC regions. The control red curve, which was generated by sequencing of unamplified genomic DNA, is our gold standard with root mean square deviation value of 0.22. Next two best curves are yellow, supplier C, and green, Equi-5, with root mean square deviation values of 0.24 and 0.29 respectively. The normalized amplification coverage evenness all over the genome is shown in this slide. In ideal case, all affected bases have to be sequenced at equal coverage of 1. However, nothing is perfect in the real world, and our gold standard red curve has the evenness value of 0.75. Single-cell DNA amplified by Equi-529 has the best evenness score of 0.5 among all tested commercial kits. Another important indicator of whole genome amplification evenness is MAP score, which describes how reliable we could perform copy number variation analysis. It is a common knowledge that MD-based whole genome amplification has better coverage overall, good single nucleotide polymorphism calling, low GC bias, but due to intrinsic nature of some hotspots in amplification initiation, it is not as good for copy number variation analysis comparing to hybrid whole genome amplification techniques like ballpark. It is not a surprise that MAP curve of Equi-529 amplified single-cell genomic DNA is not the best among tested kits. However, it is still at pretty good level and could be used for analysis. Finally, I would like to show last but not the least slide of mine, where you find two references of our collaborator, Dr. Ramūnas Stepanauskas from a single-cell genomics center, Bigelow, United States. He has used our amplified DNA polymerase in his single-cell research targeting microbial cells. Taking into account that bacterial genome at single-cell level is about 1,000 times smaller comparing to human, it is not a surprise why the data is published in Nature Communications and Cell. At this point, I would like to give a word back to my colleague, Agne. Agne, please.
Thank you, Dr. Skirgaila. It's now time for our second audience poll. Now to our audience, our next question is, are you currently or planning to develop diagnostics assay using isothermal amplification? Again, simply check the yes or no box. And we thank you for your participation. Dr. Alminaite, you may continue with your presentation.
Remigijus, thank you very much for an interesting talk. And now let me continue with a LAMP and where this kind of method can be used. So talking about LAMP, or in other words, loop-mediated isothermal amplification, first of all, what we have to say is that this method is really irreplaceable when immediate, fast pathogen diagnostic is needed. This method, because of its characteristics, offers speed, robustness, simplicity, and ease of detection. So what kind of amplification is LAMP? This method was first described in 2000 by a Japanese scientist, and LAMP amplifies DNA with very high specificity, and this polymerase is very efficient under isothermal conditions. The polymerase that carries out the reaction, DSM DNA polymerase, has very strong strand displacement activity, and LAMP becomes a standard method for isothermal diagnostics. Let's look into more detail how the LAMP reaction is set up. So there are only so many components that go into the reaction. So what is needed is the polymerase, a DNA sample that can be a poorly purified DNA even, and also a set of specific primers. Now, what I would like to point out is primer design is crucial for performing LAMP amplification. The primers have to be specific for the regions that are at certain distance from each other. 4 sets of primers, including a pair of outer primers and a pair of inner primers, have to be used, and then at least 6 distinct DNA regions recognized. And at this point, when everything falls into the place with the right primers and the specificity, the reaction can continue with speed. When DSM DNA polymerase is coupled in a reaction with reverse transcription enzymes, we can also develop methods for RNA as a starting material. As the reaction continues, we can see that LAMP is only simple at the first glance. In fact, there is a great complexity in the course of this amplification. The final products of the reaction are stem loop DNAs with several inverted repeats of the target. These cauliflower-like structures with multiple loops are formed from the hybridization between the alternatively inverted repeats of the same strand. And what is probably the best about LAMP is that the result of such reactions, due to very efficient DNA amplification, can be visualized even by the naked eye as the reaction turns turbid. Alternatively, changes in color can be detected, and when developing LAMP-based assays, even mobile applications can be used to visualize or detect the positive result. So the benefits of the LAMP method for rapid diagnostic assays is evident. Efficient amplification enables rapid diagnostics in the field. Virtually any pathogen can be detected using LAMP. The pathogens include plant pathogens, plant viruses, for example, infectious disease agents like malaria, Zika virus, tuberculosis, the list continues. The tests have been developed even for coronavirus detection using LAMP. The testing can be done outside of sophisticated laboratories, even in the field without any sophisticated equipment. This broadens the surveillance efforts that can be carried out. And this type of reaction is highly resistant to inhibitors and allows us to use complex samples such as plant tissue, blood, urine, saliva, and these type of samples require minimal processing. So when the reaction conditions are right, they can continue even with poorly purified samples. At this part, let's see where isothermal amplification methods are used in diagnostics. First of all, I'd like to talk about whole genome amplification and its use in new generation sequencing workflows. So whole genome amplification comes in very handy when nucleic acid extraction does not give us enough starting material. For example, when starting material is just a single cell. Then in the starting steps of the nucleic acid preparation, very processive DNA polymerases such as Phi29 or EqV-Phi29 DNA polymerases come in very handy for the first amplification of the starting material that can then be used in the library preparation, purification, and finally when the assay continues to sequencing and data analysis. So whole genome amplification is one of the key reactions allowing us disease diagnostics and in general research that is done with a material that comes in very small amounts. Examples of such material that we probably are more familiar with, for example, when non-invasive prenatal testing is being performed or liquid biopsy of tumor cells that circulate in our blood. On this slide, let's see where the LAMP comes in molecular diagnostics. So for this method, two very clear areas can be determined. So LAMP is widely used in diagnostics in the field, and the advantages of this method include fast reaction speed, as short as 15 minutes. Usually the reaction takes place in around 30 minutes up to 1 hour, but even results in 15 minutes can be obtained. Reaction is inhibitor tolerant, it's easily visualized, and so the challenges that LAMP reaction brings with itself is one of the most important is that it's mostly quantitative versus qualitative. So it gives us the answer yes or no. The other challenge with this reaction that it has to be developed for multiple targets. When we design the primers, it's usually to detect one pathogen. If we want to detect a set of pathogens in the same reaction setup, it's a bit challenging using this type of polymerase. However, innovation comes to this field as well, and multiple target detection is starting to be possible for LAMP as well. Another field where LAMP is applied is in portable diagnostic devices. Many such devices are now being developed to facilitate disease diagnostics in point of care institutions or even at home. It offers simplicity of operation and simplicity of pathogen detection. One can get a yes or no answer in virtually no time, and also at the accuracy that is equal to laboratory. However, this also brings in a challenge to adapt such a reaction to the conditions in a small device and get same quality. And automation for a portable device will also require lyophilized assay, which would be stable in a room temperature. And lyophilized assays can be achieved using enzymes that are adapted to Lieready conditions. So here you can see a list of Lieready enzymes and what is so special about Lieready enzymes. These enzymes have virtually the same functionality and performance over conventional enzymes, but they are formulated with a buffer containing very low glycerol content. This small concentration of glycerol will not inhibit the lyophilization, and such enzymes can also keep the assay stable at room temperature. So assays that require such temperature can be developed using exactly these enzymes. If you are looking to develop any type of assay that has to take place in room temperature, then both Equify 29 and the BSM DNA polymerase are available. Also, Thermo Fisher offers many Lieready enzymes used in reverse transcription, PCR, or real-time PCR, as shown in these tables. When developing an isothermal amplification assay or any molecular assay, Thermo Fisher offers not only enzymes, but also custom off-the-shelf plastics for your platform, or instrumentation such as thermal cyclers, and they can be even private labeled or with software customized for your specifications. And lastly, also oligos and primers, which are critical for successful LAMP amplification, for example, can be ordered for your targets, and they come in high quality. By partnering with us for products and services, you will not only get the highest quality, but also an experienced supplier that has worked with many leading molecular diagnostic companies. Here at Thermo Fisher, we understand both the critical raw materials and the new technologies that can help you develop the cutting-edge products, and we are dedicated to our diagnostic partners. So we are trying to deliver value beyond the product. If you are interested, or if you found any of the products that you'd like to know more about, please find more information on our website. I would like to now conclude today's seminar and thank the audience and also my colleague Remigijus for presentation, also Labroots for hosting the webinar, and I want to open it up for questions now. Thank you.
Thank you, Dr. Alminaite and Dr. Skirgaila, for your informative presentation. We will now start our live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions already coming in. Let's start with this one. Thank you for the webinar. I am currently using BST DNA polymerase for developing a diagnostic assay. How difficult is it to swap BST polymerase with BSM polymerase, and what is the difference between the two enzymes?
So thank you for the excellent question. To answer the question how to swap BST polymerase for BSM, or is it difficult to do? The answer is no. It's not difficult. These two enzymes are basically the same. They come from different type of bacteria of the same genus Bacillus, and they can simply be changed easily for your assay. So BSM can be used instead of BST.
Thank you. Our next question. Thank you for the very helpful content. I'm looking to develop an RT LAMP assay for COVID-19. Which isothermal amplification enzyme do you recommend for RT LAMP assay?
Thank you. Thank you again. Very important question today. So to answer the question, when developing an RT LAMP assay, I would like to recommend to use a combination of two enzymes. I would recommend to carry out the first steps of the reaction with RNA transcription, because the virus, as we know, has an RNA genome, and to use RNA target, we need to have the reverse transcriptase for the first step. So from Thermo Fisher portfolio, that would be a superscript 4 or Maxima, and then go on using the BSM polymerase. At the same time, if the conditions are worked out, or after the reverse transcription amplification step, and using BSM, the LAMP reaction can give you a quick result and visualization. Just to remind that both of these type of enzymes, the reverse transcriptases and BSM, comes in Lieready format, or are available in Lieready format, and RT LAMP assays for COVID-19 can be developed for ambient environment.
Thank you. And we have time for one more question. I need to perform WGA on rare cells obtained by microdissection from FFPE tissues. Do you know if Equify 29 has been used for WGA on this kind of sample?
Yeah, so actually, very interesting question. So nucleic acids extracted from FFPE samples are heavily fragmented and typically are too short for efficient amplification by standard MDA reaction driven by Phi29 or Equify 29. So that for special workflow would be recommended to generate long amplicons before MDA reaction to proceed.
Thank you. Now, before we go, I would like to thank our audience for joining us today and for their interesting questions. Those questions we were unable to answer live today, and those submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. I would like to once again thank Dr. Agne Alminaite and Dr. Remigijus Skirgaila for their time today and their important research. We'd also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and Labroots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon.